our special guest, and it is my pleasure to introduce today's special guest speaker, Dr. Cristina Correia. And Dr. Correia is assistant professor of the pharmacology in Mayo Clinic with extensive expertise in system biology, cancer genetics, and the AI-driven computational modeling. Especially now, another emerging model system is NAM. You may heard about NAMES. That is also another hot topic. And Dr. Correa's involvement is really necessary component for this new emerging study. Her work focusing on the integration multi-omic data to understand the complex biological system and identify therapeutic target. And she has developed several innovative computational platforms, including network-based machine learning approach to analyze genomic, transcriptomic, and the proteomic data. And her contribution include tools such as net decoders and more recent AI-based framework that enable the discovery of the disease mechanism and also predictive biomarkers. And Dr. Correia's research has significantly advanced in our understanding of the cancer biology, particularly in the areas such as drug response, genomic instability, and the network-based identification of the targetable pathway. And her work spans from large-scale data integration to translational application in precision medicine. So today, Dr. Correia will present the integrating system biology and the AI, highlighting how computational and the AI-driven approach can be combined with the experimental system to better understand the model complex and the biological procedure. For questioning, you can turn on the mic anytime to speak up or post the question at any time in the chat box. And please join me in welcoming Dr. Cristina Correia.
Thank you, Dr. Kim. It was an amazing presentation and introduction. And so I'm going to start with my presentation. Let me bring up my slides. Can you see it? I'll put it on slideshow. Can everyone see it?
Yep, great. You can start.
Perfect. So today, I'm taking this presentation a little bit on the educational side to bring like key ideas to take into account when looking at the diverse multiomics data. And the title of my presentation is AI-powered medical discoveries. So in this slide, I'm really focusing here on, so I work closely with Dr. Hooley at Mayo Clinic. We're looking at the AI-driven ecosystem. And why are we interested in AI-driven ecosystem is because all diseases are complex, they're heterogeneous, and they're all different. So our aim is to build systems AI tools. They're in principle agnostic, and then tailor them to a specific disease. So today I'm going to walk through some of those tools. And initially, I just wanted to highlight that we use data from preclinical models like mouse, organoids, cell lines, stem cells, as well as clinical data from patient cohorts. And so the tools I'm going to be talking today is going to be net decoder that is shown here. And we're going to be building networks. I'm going to talk about differential utilized networks and how they can be used to think about phenotype engineering. Next, I will go into Perimeter. So it's a tool that is thinking more instead of a cohort-based analysis, is like individual patients and variants within those patients, and how can they be used to define context-specific therapeutics. I will also touch base with Spin AI, which is a tool that was developed for spatial transcriptomics. It's really emerging. A lot of data is coming from spatial transcriptomics, has a lot of possibilities. And we're going to talk about spatial predictive genes that allow us to identify the location of cells within a tissue. And then there will be another tool that I'll present, it will be later, but then I'm going to wrap up with what's different AI and thinking how we form complex and algorithms to put all this information together and really thinking how we bring new knowledge to the table. And overall, our goal is really to better improve outcomes for our patients. So I think I'm just going to start with systems. I think everyone is kind of familiar what is a system. A system is made of many components that interact with each other and form complex webs. And the system itself is more than each individual part separately, and combined together gives a higher emerging properties. So our goal is to build hypothesis-driven tools. And for that, we use models to understand how a system works. We test hypotheses with our models. We compute the model and do the model using a variety of dimensional data. We ask questions from our trainer model, and our goal is to retrieve mechanism insights and validate them with preclinical and clinical data. So the first one I'm going to talk about, then I'll go slow, or I'll go. I want to really put some principal key concepts here. Is NetDecoder. So NetDecoder, the goal of it is a phenotype gene prioritization tool that looks at different phenotypes and determines what drives those phenotypes. And it's a modeling tool that looks at process-guided flow, and it models the flow from a start to an end. And you can think about a faucet that you turn on the water, and you're going to see where the water is going to drop. So to start with NetDecoder first, to explain how the way actions it works, I'm going to focus here on this main panel that shows what we call a network or a graph. And this graph is composed of nodes and of vertices. They, in this case, are codified by these different shapes, and I'll come back to that. And then there's edge or lines that connect these different nodes. And these edges in this particular case are going to be protein-protein interactions. So we're thinking about the protein-protein interaction network, where we have the nodes are proteins or genes, and the edges are their relationships. How do they interact with each other? So the reason why they have different shapes is because they have different properties. So we can find different properties within a network. And so, for example, here the diamonds represent genes that we consider the sources, which is the genes where the model is going to start modeling from here to the end, like as I said, as water dropping, and it goes to the transcription regulators that in this case are the squares. So another thing is that the nodes within the networks will have different properties, and I'll come back to that. So the way the NetDecoder was designed is a network tool that looks at different phenotypes. It could be phenotype 1, phenotype 2. This phenotype could be normal or disease. It could be a drug treatment. It could be a knockout. It could be a CRISPR knockout or a knockdown. And it could be a drug treatment, as I mentioned before. It could also be a perturbation that we do to the system. So we use that to model our phenotypes. And we want to understand how phenotype 1 is distinct from phenotype 2. It's also a tool that looks at cohorts. When I'm saying cohorts is more than, I'm saying approximately 10 or more samples, and with each group. And so the way we start, as I mentioned, we have a protein-protein interaction network that I define it here. There are several databases that have protein-protein interaction networks that we actually use as our starting point. For example, BioGrid, String, RF Index, we use RF Index, and we look at these protein-protein interactions. They have approximately 80,000 genes, more than 200,000 interactions. And so one thing to know about these protein-protein networks is that they are pretty disease agnostic, and they're not specific to a disease context. So our first step to create a tool and think like it's an agnostic tool, but how do we make it specific to a specific phenotype that you have interest, for example, Alzheimer's disease or Parkinson's disease or other system that you focus on, is to use the transcriptomics, and then we overlay the transcriptomics within each of the phenotypes within that protein-protein interaction network. And we focus on genes and interactions that are detected within our data set. And that way we bring context specificity to our tool. And then we can use, for example, differentially expressed genes, or we can think about mutated genes or drug targets or any other information that we want to model. And so the modeling will start here, as I mentioned before, from the source to the regulator, and we compare the two different networks. The reason why we have different colors is because they also give us the directionality. So the outputs from this tool are, for example, the edge flow. So, for example, is the comparison. So, an edge will have a gene A and gene B and compare how do they perform within the different phenotypes. Additionally, we can think about network routers, which is one node that will have a lot of outgoing interactions, or a key target, which is a node. In this case, is a transcription regulator that has a lot of income information that comes to that specific gene. And then we can compare gene A and gene A within the different phenotypes when they have different impact genes, meaning the number of interactions that they have changes significantly between the two phenotypes. Additionally, we can also build up prioritized networks. So we'll have a network for phenotype 1, a network for phenotype 2, and we can have one that is distinct, the difference between the two. So the key message from here is what is a network? How is it composed? How do we make context specific? And also very importantly, we're not just thinking about differential expression of one single gene within a phenotype, but we're thinking how the gene acts within an entire network and how it relates with its neighbors. And so from here, we get, for example, from a normal transcriptomics data set, we can retrieve quite a lot of information that can be used downstream. And there could be GSCA and other tools that we can look also at pathways that are altered within this specific phenotype. We have used net to code in cancer as well as Alzheimer's disease. And we can also think as a way to target specific nodes, how we target the specific node. And that's the reason I was saying can also consider be a perturbation tool that we can think how do we target with a drug or a mutation or the others and what is impact from these two. So a concept that I mentioned, and I think this is kind of like just a take-home message, is we think about networks, we think about genes in combination with other genes, and the modeling allows us to think about we have routes that can be highly used and others not, and that gives us the importance of a gene within a network. Next, I'm going to move to a different tool that is Permuter. So in the previous tool I talked about, it was cohort based, and we use the PPIs as our starting point. And from that PPI, which is the protein-protein interaction network, we make it specific to a disease. So the problem that we wanted to solve here is different. We focus on individualized disease. This was defined in particular for cancer, but we've been applying to other disease for like obesity or complex disease. And so the idea here is that we have a lot of information, but most of the times when we think about a cohort, we're focusing here on this section of the figure that is high, like he has these dotted lines. So we think about components or features that are common between the different patients. However, there's a lot of information that we discard that could be useful and it could be interesting for targeting, also thinking about individual patients. So that was kind of like the backbone of Permuter, is thinking about individualized disease, think about cancer and how mutations can cooperate and how we can better understand those patients and target those patients. In terms of strategy, Permuter is also an integration platform. The reason I say that is because we can use normal tissue RNA-seq, we can use tumor RNA-seq, and then we can use genomics. It could be whole exome sequencing, it could be the whole genome sequencing, it could be amplifications of that information that is available. And in this particular case, to prototype Permuter, we focus on breast cancer that had 90 patients. So the key concept I talked before was what is a generic PPI? So a generic protein-protein interaction. You have the nodes, you have the edges that are the lines, and you have an interactome for a specific disease, for a global disease. And so this has 15,000 genes, approximately 200,000 interactions I mentioned before, but we want to make specific for a patient. So our strategy was to, number one, use the fold exchange, so we comparison between normal or tumor. We used information of the patient in terms of genes that are mutated. In this particular case, I'm referring to cancer, though we think about missense mutations or nonsense mutations, and mutations that have impact on the disease. But if you think on a complex disease, it'd be others that have a higher frequency in certain phenotypes, so others. And then we can use what is really important is known disease drivers. So we have a lot of information. It has been collected across many different studies, and we can use that information to augment our tools. And so in this particular case, we use genes that involve in cancer, their GDC database, that they have been classified in different tiers, and we can say this is a tier that we're focusing on. And so with all this information, then we can apply the shortest path identification that is going to allow us to connect the different dots from the things that I mentioned previously. And so we can form a lot of different short paths across the patient, and then we assign a random distribution and apply a cutoff with 100,000 random paths to see which ones are the most significant. So at the end, we build an individualized disease network, which is different from NetDecoder because it was more a cohort-based network. And here's kind of like a schematic diagram on top that we have just to, we want to give the principle of how important is this gene for one patient versus the other. So for example, here you have in the blue, you have this particular gene is very important for patient blue, but it's not important for this patient. And for example, this variation, mutation is important for patient yellow, but here it does not have any impact. And so the outputs for the Permuter tool is what is shown here in figure A and figure B. And what we have, I'll walk slowly through that. So we have a network that shows the interactions between the different genes. And the genes are the nodes, the color with the frequency that they're detected in TCGA. And the size of that node is based on the Permuter importance. So Permuter importance comes from the previous slide when we define the paths and say this is an important path and this is how much that specific mutation contributes to that path. And so what I think it's important to highlight here is that we have two different patients, both breast cancer patients. They have here in A, we see a more sparse network, number one. Number two, we have PI3 kinase is highly contributing to this network and driving a phenotype. However, in patient B, that is not the case. We have a very dense network that has many different mutations. PI3 is also identified, but it has a totally different weight for the network. So this really gives you an idea, how can we use information that is available, that is not commonly used, that we can use to define single patients. Additionally, we can also use this information. I don't have a figure here on that, but in the paper, and I have a reference for the paper here, is that we also use this information to match drugs that can better tailor to a specific patient. And so that is really interesting when we have, for example, the kinase or others, and also think about different drug combinations that can be used. Jumping to AI, more specifically. So far, some of the key concepts is like it was networks, and we had predefined networks that we use protein-protein interaction networks that are available in the public domain, and we tailor those to a specific disease. N takes a different approach. So N stands for artificial neural network encoder, and it uses an autoencoder to define what are the key features for a specific phenotype. But more than that, what it uses is it opens a black box, and we use what we call late engineering methodology to determine the weights that are used within the autoencoder to build our specific networks. So the way it works is that we have a cohort of patients. We have, for example, not special transcriptomics. We have transcriptomics or single cell. We'll feed that to our encoder and determine the features there, determine the separation between two, for example, two different phenotypes or two different subgroups. And then we can go back and say, let's extract the weights of what was learned during this process and build our de novo networks. So it's a totally unbiased approach that builds networks that are specific to a specific phenotype. And so with this, we can form like what we call bring the opaque predictions. We can open end and start thinking about how can we really bring mechanistic insights. And it's another key thing I want to think about is like when we think about AI and other methodology we use in number one, there is not one tool that fits all, but we really can use our knowledge to bring into our methodologies to get more mechanistic insights. I'm jumping into Spin AI. So Spin AI is spatial predictive artificial intelligence. And so I mentioned before, we using spatial transcriptomics in tissues as well as I think they start also doing in some organoids. But what we thinking here is how can we define what determines a specific tissue architecture? So the basic idea between Spin AI was to identify this class of genes that inform where cells are located within the tissue. So we started with the tissue. Here, for example, I have a section of the brain that was used for spatial transcriptomics. Spatial transcriptomics, you're going to have beans or spots. And I'm thinking about, sorry, StereoSeq or Visium or other technologies that can be used for spatial transcriptomics. But you're able to collect quite a few transcripts within each of those spots. They can be single resolution or not, and we can use an unbiased approach to detect where are those genes located and what are the genes that infer that location and contribute to a certain phenotype. So here's a more schematic representation of what we do. We use spatial transcriptomics. We did a cross-fold validation here just for shown, and that is our input for our deep forward network. And we have this split between train and test. We train our deep forward network across the different folds, and then our target goal is to predict the XY coordinates. And because we use different folds, for example, it's the same slide, but just the data is split between different folds. We can aggregate the data and find combined predictions and determine what's the genes are the most important for a specific region within the spatial transcriptomics. And so this was prototyped with a published paper, but we've been applying this to cancer as well as other diseases. And I think there's a lot of potential in spatial transcriptomics section of field. So I'm kind of getting to the end because I left a lot of time for also questions because I just want the key concepts to be like really crystal clear. And we're thinking about the pulse-driven AI. So as I alluded before, we're thinking about how do we embed, how do we collaborate humans and machines, and how do we bring the most out of it? And so really what we want to do is use all the knowledge that we have ourselves and all the knowledge is available in the public domain and embed that within our AI models to better test hypotheses and predict which gene and associations are important for a certain phenotype. So we call this a hypothesis-driven learning algorithm, and it's a new type of AI that we patent 2024 that we've been working on. That is, how do we incorporate all the information that is out there, pathways and other things within our models to then get more interpretability of these models too. And so to finish up, I think I gave a couple of examples of how to showcase the concept of hypothesis-driven AIs. Like how do we have hypotheses that we include within our methodologies and get the most information out of it. And also thinking how we perturb different systems to now understand how they're going to impact phenotypes or other things that we do downstream. So it's a new kind of dialogue between the human and machine that we really want to do this, what we call thought experiments of doing in silico experimentation and perturbation to understand how systems work. And kind of like, I'm open to questions. I hope you guys have lots of questions.
Okay, thank you for great presentation. And for the question, please feel free to raise your hand or post your questions in the chat box. And we will have a question and the answer session at around 10:50 a.m. PST. And okay, good. And also, I have just one quick question. One very really interesting emerging concept is idea of the biological digital twin,
Mhm.
where AI and the computational model stimulate human disease or even individual patients. Do you think stem cell system and AI are getting closer to making this kind of things really realistic in research and also clinical field? And also another similar question is, what is the biggest challenge right now?
Mm. Okay. I think both are great questions. Number one, yes. When we think about the ecosystem that we're creating like that digital twin and trying to use different perspectives to bring the data together to answer questions and to monitor. So digital twin, the key feature, one of the key features of digital twin is thinking what is happening now, but we really want to infer what's going to happen next. So that's the one of the challenges. And we thing or what we propose is that we can use different ways of looking at the data, and especially thinking that's why I was kind of going back and forth with the perturbation, how we perturb and can predict what's going to happen next. So it's kind of, I think it's a big area of investigation. I think another key challenges are what many have referred to before is the dimensionality of the data, but most importantly is how the data is sparse in some fields, and also how is it disconnected. And so we need to emphasize this connectivity between the different sources.
Hey, great. The next question is from Dr. Omar Farah.
Yeah, thank you, Christina. That was wonderful. Really, really great talk. Kind of related to Kitai's question, and your answer actually. I was wondering how negative data being underrepresented in the literature. How do you account for that? How do you fix for that? I think one of the guest lecturers last week was actually talking about how they're starting a special edition of a journal that is dedicated to negative data specifically. Is that a concern, or is it not?
So
What,
I,
when is that?
I really like your question too. I think it's a very interesting question because even thinking just, a basic concept of the networks, we there's always biases, right? There's bias for what's reported, there's bias for things that are over-reported, if bias for things that are not reported, right? So how do we combat that? One thing is like, for example, having resources say this does not work or this work or we see this and that, right? Or kind of go through that. It's a kind of an iterative long process. I think what we try to do too is like always think about how the way things are done in the wet lab. What's our negative controls? How can we use things that we know they don't work or they should not work as a way to refine our tools? And that's kind of one of the approaches that we're taking is like, let's think like, for example, if we've been thinking breast cancer, let's think of a totally different cancer that doesn't, it should not be having the same phenotypes, different tissue, everything, and use that as negative. I don't know that we have more than that right now. We also look a lot at different resources. So that's one thing that I did not go into detail, but there's a lot of public available resources like GTEx and others that looks at normal. And right now they're focusing mostly on there's a certain bias for the population that initially gave the sample, so it's like 50 plus or 45 plus, but there's a big interest of doing longitudinal, right? So the longitudinal, which goes a little bit to the digital twins, is like, what is the age? I mean, there's a lot of different factors that can contribute to that, but thinking like how we can use the normals as a way to think it doesn't happen. It's still not a negative like you're saying, but it's a starting point.
So, yeah. And I had another question around, again, maybe a little bit related, but on if you could elaborate a little bit more on some of the assumptions made around how the impact of gene expression for a particular gene, or let me rephrase this, the impact of differential gene expression in its like in an endpoint review of the impact in a network, right? So for example, in development, sometimes the genes that are most closely guarded and regulated are the ones that would have the biggest impact if turned on, so that they would only be turned on for a short period of time, or expressed very, very minimally differentially. Is that considered as part of the AI models that are being built, the axioms in which these parameters are modeled?
I think that's also a very good question, and I think we tried to address that in Netcode, even though it's an earlier tool, but a lot of the concepts migrate to the new tools that are being used, right, that we're building. So the concept in Netcode is that differential expression is not really what might drive all the phenotype, right? Because there's many there's many regulatory processes that occur in between, like protein, RNA, etc. And then you're thinking like a little bit of transcription factors that have a very defined role of expression, etc. So by thinking about the network, we're not focusing necessarily just on expression, but what everything contributes to it. So if you see a lot of the targets that change, then you'll be able to identify it. So it kind of overcomes a little bit that. So there's ways, it's kind of like a little bit of reverse engineering that we're thinking of.
Very cool.
Yeah, I have another question. And this question is just a general question in the future AI and the computational biology. And Dr. Correia, as AI model become more powerful, do you think future biological research will shift more toward the hypothesis generation by AI, just rather than just a only human-driven experimental design? How should young researchers prepare for this kind of transition? And this one question is could be very broad, but this one is could be very good advice. How young scientists will prepare for this kind of hypothesis driven by AI, and while
Mhm.
still maintain the strong biological understanding and the experimental skill.
I mean, I don't know if I have the answers to that question because it's something that is in the public domain. Everyone has their own opinion. So, what I can express is that number one, we can learn how to navigate AI. And I think there's a lot of pros and favorable things that we can get. We can get, faster information. And I think really what we're trying to do is this concept of the interactions between the two, between the human and the machine, which is like illustrated here with the hypothesis-driven AI. Is like, okay, we have this idea, this hypothesis. Let's quickly try it in a test, like in a wet lab. No, sorry, in the computer. So we kind of perturbate the system, see what comes out of that perturbation and that prediction, and then say, okay, is this a reasonable or not output that we can test in the lab? So instead of taking like, one gene at a time, maybe we're doing a lot of computation at the same time and say these are the key genes, and then you go back and more quickly test if that is the case or not. And it also is a way to validate what we're seeing in the computational side in the models. So I think it's really important to understand both and think like how we can benefit and you know really put the two together to find our key questions and even ask questions that we have not thought about and say, how do I validate these? So I don't think validation is ever going to go away. There's changes, there will be ways that we work differently, but it is important. And I think knowing how to ask the question, understanding, all the key factors that drive a specific phenotype of system is really important. So I would suggest immerse yourself and take the best of both worlds.
Thank you very much for all questions and the answer. And then also Mayo Clinic Computational Group is our center's strong collaborative partner last more than 15 years, and we publish together with more than a dozen papers. So therefore, I really appreciate your participation, continuous collaboration. And anyone is interested in this kind of collaborative work, and then feel free to contact us and then develop the new idea, new project, or grant idea. We can submit grant together. Thank you, Omar, Tom, and Dr. Correia, and for the excellent and insightful final lecture. And then today's session highlighted how neural differentiation system include both 2D and 3D model advancing through the integration with the system biology and the AI-driven approaches. So this concept are broadly applicable and they increasingly important for understanding complex biological system and improving just the translational outcome. So before announcing my closing remark, do Tom and Omar have any closing comment?
Omar, you want to
Yes,
go
for
or you want me to
sure.
go?
Yeah, you can go first, Tom.
Fantastic. Well, I would say just as a whole on the course, first of all, thank you all very, very much for joining. We hope it's been interesting. We hope it's been helpful. And this is something that we've done 2 years in a row now, and that we are continuously trying to improve. And so any and all feedback that we get from you guys would be massively helpful in that, with regards to content that you saw, with regards to content that you'd like to see, with regards to the way that we can improve the course. I don't want to speak for the other instructors on the course, but Omar and I are certainly massive nerds and very, very passionate about the space in which we work, the science in which we get to explore. And I mean, it's such a freaking cool space. Stem cells, neurobiology, cardiac biology. I mean, you name it. We are in such a hot topic space at the moment, and we want to translate that excitement through to you guys. I probably can't even count the number of times I said, this is really cool while I'm talking. And I'm like, oh, I must sound like such a massive nerd. But we really appreciate you guys joining the course. We hope our enthusiasm is at least infectious. And we're going to continue doing this course through our collaboration with UCLA and CERN. So please feel free to reach out if you have any feedback that you'd like to give us. But thank you all so much for joining.
Perfect. And I'll just echo that. Thank you massively to everybody, your participation, your questions. As Tom mentioned, we love doing this. In fact, you might receive a survey to fill out afterwards via email. If you guys could please take just 5 minutes to fill it out, I promise it won't take you longer. Just to give us, again, as Tom mentioned, pointers on what you liked, what we could change, and how we can make this better, right? But as always, now and in the future, please feel free to reach out to myself and Tom. Amongst any and place us amongst your resources if you need any help. And my last hurrah here, honestly, be to try to be as collaborative as possible, right? Our field has stagnated a bit in terms of this was a while back in terms of either funding or ability or access to resources, reagents that we can work with these cells pretty well. And now we're at a really good point, right? We don't need to reinvent the wheel. There's a lot of know-how. So please feel free to reach out, collaborate, use your resources. Things like your course, for example, with Kitai, just massive resources that are at your disposal, excuse me. So please feel free to use them, right? And with that, again, thank you all. Hope this was as much fun as it was for you as it was for Tom and I.
Thank you. I would like to thanks for all participants for your active engagement throughout this program. And over the past 7 weeks, we have discovered key topics, meaning like stem cell biology, culture system, genome engineering, and then differentiation strategy. And as mentioned, the participants who attended at least 6 lectures will receive certificate of the completion of this training. And some of you joined the program, right? And you can take the missing section next year to get the certificate. And also additional information regarding certificate and the access to lecture material will be shared by email. And all of the lecture recording will be including the special guest the presentation will be uploaded to our center YouTube channel after course is completed. And I would also like to acknowledge the support from Thermo Fisher Scientific as well as our UCLA Human Stem Cell and Genome Engineering Center team member, including administrative and technical staff who made this program possible. This fall, we aiming to organize another educational program focusing on another emerging robotic automation tool that enable to large-scale sample processing and automated data collection at the 1,000 or even 10,000 of the sample level. Usually we cannot do with the human hand 10,000 sample data collection procedure, not possible. Only robotic automation and the AI or industrial partnership is always make this kind of new ideas possible. And also you can design the unique project that others could not without this kind of robotic automation before. And this program will be organized in collaboration with the industrial partner. This education program is open to level, and the additional detail will be posted in soon. Not only with just providing the core service through core-to-core partnership with your institution, through your partnership core facilities such as animal core, stem cell core, gene editing core, or some of them is organized core, you can access to our core service. But also if you are also interested in the independent research, collaborative research with the robotic automation in our center, working together with our special guest and the industrial partnership, combining robotic automation, bioprinting, AI incorporated or automated data collection and more, please feel free to contact us. And we can initiate to idea development and then grant writing, anything as a scientist to scientist. We open our mind and then make the great project and then also share the benefits together. Thanks again for your participation, and we look forward to future collaboration. Thank you.
Thanks, everybody. Have a good one.